Hi class, this is your instructor, Skylar Huff, and I'm here to review things for your fourth test. So I'll be reviewing for you all chapters 9 and 10. Let us begin. So as I do begin, one thing that I will say is please make sure you have your lecture notes out. I say again, please have your lecture notes out. And I say have your lecture notes out because you need your lecture notes to review. And along that same note, by having your lecture notes out, just please keep in mind that you should add to your notes, write down any questions you may have, and of course have your textbook there and the presentations for these associated chapters. Let us now begin. So in this chapter, we begin with structure of skeletal muscle. So as I begin with skeletal muscle structure, please keep in mind that skeletal muscle, as it is, it has layers of connective tissue that enclose it and separates all parts of that skeletal muscle. So having said that, keep in mind that the individual skeletal muscle is separated from the adjacent muscles and held in position by layers of dense connective tissue, and this is called fascia. So the connective tissue that surrounds each muscle may project beyond the ends of the muscle fibers and form that cord-like tendon. So those fibers in a tendon, they may intertwine with those in the periosteum of bone and attach muscle to bone. I say again, attaching muscle to bone. So with that, the layer of connective tissue that I refer to now is going to be that closely surrounds a skeletal muscle is called the epimysium. The epimysium. So you should be looking at the diagram we drew in the lecture. So in some areas of the body, it may merge with the surrounding deep fascia, and we have another layer of connective tissue called the perimysium. So the perimysium, it extends inward from the epimysium, and it separates the muscle tissue into small sections. Those sections are called fascicles or fasciculi. So each fascicle is individually wrapped and bound by what is known as the perimysium. So each muscle fiber within a fascicle is further compartmentalized. And here I'll refer to that being referred to as the endomysium. So I say again, the endomysium. So it is that endomysium that surrounds those individual muscle fibers. So now we've done each of those. What I'll now get to is describing muscle itself. And as I describe muscle now, I'll begin here with its basic or general characteristics. So there are three types of muscle, which this should be very well reviewed for you all. In those three types of muscle, I refer to them as being skeletal muscle, cardiac muscle, and smooth muscle. And as I've done those, I'll now get to skeletal muscle. It is attached to bone. It covers the skeleton. It has long cylindrical fibers. I won't necessarily call those cells, I'll call them fibers. It is striated. And to not go too fast, I do say those fibers refer to skeletal muscle cells, and it's because they have a fiber-like appearance. Additionally, they are multinucleated, meaning each cell is multinucleated with the nucleus right beneath the sarcolemma, that cell membrane here in muscle. So I'll continue where I left off by saying that skeletal muscle is striated and voluntary. It contracts rapidly, however, it's easily tired. So skeletal muscle is a rapid contractor, meaning it contracts rapidly and tires easily, and it is tremendously powerful, yet adaptable. So when I say it's adaptable, I say that you can pick up a 6-pound book, the textbook you have, and then contrast that with picking up just a small little paperclip. That is just how adaptable our muscle fibers are. Up next is cardiac muscle. Cardiac muscle is found in the walls of the heart. It is also striated, however, it is now involuntarily contracting. So I say again, the heart muscle is involuntary in its contraction, and it does so without nervous system stimulation. It's so amazing how it does it. So it contracts at a steady rate and is set by the heart's pacemaker. So yes, the heart itself has its very own pacemaker. So although there is neural controls, those neural controls can speed up the heart for brief periods of time, such as with running. Thirdly, smooth muscle. Smooth muscle is found in the walls of hollow visceral organs. Those are things such as your respiratory passages, the urinary bladder, and the stomach. So it forces fluid through internal body channels. It has elongated cells, and those elongated cells are referred to are involuntarily contracted. So cardiac muscle as well as smooth muscle are involuntary, and its contractions are different. So the contractions here are slow and sustained. I say again, contractions here are slow and sustained. So it's called visceral smooth muscle. So now I'll get to basic characteristics of muscle. Basic characteristics of muscle itself. So to begin with the basic characteristics of muscle, what I'll begin with now is going to be that number one is muscle is excitable. I say again, skeletal muscle, smooth muscle, and cardiac muscle. It is excitable. By stating that it's excitable, meaning it's responsive, it receives and responds to stimuli by changing its membrane potential. So in muscle, the stimulus is typically a chemical. Or I repeat, in muscle, the stimulus is typically a chemical. I refer to as being a neurotransmitter released by a nerve cell. With that, I'll say muscle also has contractility. So with contractility, I mean it has an ability to shorten forcibly upon adequate stimulation. Yes, it has to be adequate stimulation. And of course, that is what sets muscle apart from all other tissue types. Next, I'll get to number three by saying that muscle exhibits extensibility. That is, its ability to stretch. So muscle can shorten while contracting, and of course it can stretch when relaxed, even beyond its resting length. Excuse me. And upon stretching when relaxed beyond its resting length, the fourth and final characteristic I'll say is going to be muscle's ability to exhibit elasticity. So when I say elasticity, that's muscle's ability to recoil and then resume resting length upon stretch. I'll now get to muscle functions. So with muscle functions, I'll say that muscle functions to produce movement. So with movement, I'm referring to locomotion and manipulation because of skeletal muscle. So blood circulates throughout the body, and it's because of that rhythmic beating of our heart by way of that cardiac muscle. And smooth muscle in the walls of blood vessels, which help us to maintain blood pressure. And this also includes the digestive, the urinary, and reproductive systems. So now I'll get to muscle being able to aid us in maintaining posture and body position. So the skeletal muscles we have an amazing ability to allow us to maintain the posture that we do indeed have and the position of our body. Excuse me. And then lastly, muscle can function to stabilize joints and generate heat. So those are two separate in themselves, but of course in generating heat, muscle maintains our normal body temperature. So this has been the general characteristics of muscle. So now I'll get to what is next called muscle fibers. And I'll deal here with skeletal muscle fibers. So each muscle fiber is formed from an undifferentiated cell that fuses during development, resultant of which the multinucleated muscle fiber is a thin, elongated cylinder with rounded ends that attach to those connective tissues associated with the muscle itself. So now our terms. I hope that as you all begin every chapter, as you all began this chapter, you used understanding words. So the sarcolemma would be that cell membrane. The sarcoplasm will be, of course, the cytoplasm here in muscle. The prefix myo should make you think about muscle. So when I refer to the myofibril, that's that long parallel structure, meaning it's these groups, the myofibrils that are, those overlapping parallel groups of thin and thick filaments that allow muscle to contract. So in addition to those terms, I'll provide another term, and I'll call this the sarcomere. So the sarcomere is that functional unit of muscle contraction. So I'll just take my time here and get to the sarcomere. We drew this in lecture. The sarcomere is composed of a number of components. I'll begin by saying that in the center of the sarcomere is what I call I'll go over the M line. M as in Michael. So the M line is what holds the myomycium molecule in place. So you find the M line between the H line, and of course, what I'm getting at is we have the M line, and going outside from the M line is the H band, and then going outside from that would be the A band. I call it the A band because it's the anisotropic band, and light here has been polarized twice, hence the A band appears to be dark. It looks darker because of such. The I band called the isotropic band, and it's light because it's polarized once. So as you look at this, within the I band is what is called the Z line. The Z line. So the Z line is there, and with that Z line, you would have your thin filaments. I repeat, with that Z line, you would have your thin filaments. So having done such, what I'm getting at here with those thin filaments, class, is that you have within which the actin attached to the Z line, and we have the myosin attached by way of the M line. So going from Z line to Z line, you have what is called the sarcomere, that functional unit of muscle contraction. So I'll now get to other components. So two other types of proteins here are called troponin and tropomyosin. So I call troponin as it is that component that is there for the purposes of contraction. So troponin is here, and as I get here to troponin, we have troponin being made up of three parts. So we have troponin C, or TNC, we have troponin T, also known as TNT, and troponin I, called TNI. Having gotten to each of those, the way it happens is by way of troponin that's here for us, class. TNC is what will bind to calcium. Calcium is a foremost requirement for contraction to occur. So as calcium binds to TNC, called troponin C, it will roll away the blocker called tropomyosin. Remembering, tropomyosin is what blocks the active sites on actin, which of course would be where the myosin heads will attach, forming a cross bridge. TNT is for, of course, troponin and tropomyosin, and TNI is how it inactivates. So please again, keep in mind that tropomyosin, as it is here, it forms to block the active sites on actin. Next up, I mentioned moments ago, myosin. So it is myosin, connected there by way of that M line, is there to bind to the active sites on actin to form a cross bridge. Next up, are the transverse tubules, or T tubules. So the T tubules are here, the transverse tubules. And the reason they are here is to allow those nerve impulses to get deep within the muscle fiber themselves. So the T tubules, they come in at the border of the I and A bands and the skeletal muscles. So I call them T tubules or transverse tubules because they, again, extend into the sarcoplasm as an invagination, and they're continuous with the sarcolemma and contain extracellular fluid. So each T tubule, each transverse tubule, lies between two enlarged portions of the sarcoplasmic reticulum called cisternae. So you have the T tubule, the transverse tubule, and two terminal cisternae. They form what is called the triad. So the triad, of course, is that T tubule with those two cisternae, the cisternae on each side. So going back to the sarcoplasmic reticulum, please keep in mind that it is the sarcoplasmic reticulum that is the place at which calcium is stored. So the calcium, as it enters in, thanks a lot to the change in membrane potential, meaning depolarization, is what causes the release of calcium to come in, which is that foremost requirement for contraction, and then of course allows troponin to change shape, to roll away tropomyosin from, of course, the active sites on actin, and then allow the myosin heads to attach to the active sites on actin and form a cross bridge, pulling, of course, those actin filaments nearer and nearer to that M line, to that A band, and to shorten, of course, the muscle fiber itself. So as I continue, please make sure you all review these figures and what we've just done via your textbook. So I'll now get to muscle contraction. So with muscle contraction, please bear in mind that on your lecture test, you will describe the events occurring at the neuromuscular junction. So a muscle fiber contraction is a complex interaction of several cellular and chemical constituents. The result is movement within the myofibrils, in which the filaments of actin and myosin slide past one another, shortening sarcomeres, and when this happens, the muscle fiber shortens and pulls on its own attachments. The neuromuscular junction. So as it occurs, we must have the events that occur at the neuromuscular junction. So to get a contraction, the muscle fiber must become activated. Hence, there has to be a change in membrane potential. So let us begin. So each neuron has a process called an axon. The axon, of course, extends from the cell body, attached to which are dendrites. Those dendrites are the receptive region. So as the dendrites receive some stimulus, that stimulus then, of course, travels down, and it gets to what is called the cell body, the axon hillock, and then ultimately the axon itself. So as this happens, you all, please keep in mind the process. What must first occur is a nerve impulse. So the action potential arrives at the axon terminal, at the neuromuscular junction. As it occurs, what has to happen is that change in membrane potential. So at rest, the cells are polarized, meaning the cells are slightly more negative inside than outside. So the outside of the cell is more positive than, of course, that more negative inside. So as that action potential arrives at the axon terminal, it's a change in membrane potential from what was once resting membrane potential to now being, of course, the action potential. That change in voltage causes the voltage-gated calcium ion channels to open. As those voltage-gated calcium ion channels open, what then enters in is going to be calcium. So calcium rushes into the cell, and because of the influx of intracellular calcium still here in the nerve, still here in the motor nerve, will be vesicles of a neurotransmitter begin to form. And in this case, it will be vesicles of acetylcholine form. As those vesicles of acetylcholine form, they will then fuse to that motor end plate. meaning they will fuse with that neuron, and then acetylcholine will be released from that nerve, and then it'll begin to diffuse across the synapse, or that synaptic cleft. As that occurs, I say the acetylcholine molecules will then bind to the chemically gated sodium ion channels. I say again, they will bind to their receptors. The receptors I here refer to are the receptors on the sarcolemma. So these receptors are those chemically gated sodium ion channels. So what happens is the chemically gated sodium ion channels open and of course allow sodium to diffuse into the muscle cell. As sodium diffuses into the muscle cell, it begins to change that local voltage, meaning providing that end plate potential. That greater influx of sodium is what causes that local depolarization event. So this begins to open voltage-gated sodium channels in neighboring regions here on that sarcolemma, allowing even more sodium to enter, further depolarizing the sarcolemma. As this reaches threshold potential, this will then begin to, of course, propagate the active potential. So here, the events of this will cease, but only when, of course, the neurotransmitter has been either A, diffusing away from those chemically gated sodium ion channels, B, it is broken down by acetylcholine esterase into acetic acid and choline, or of course it just is reuptaken by that motor neuron. These are the events occurring at the neuromuscular junction. So next of which, I'll get to the way in which contractions occur. So this gets into the excitation-contraction coupling. And it's otherwise stated there as being that sliding filament model of muscle contraction. So for this to occur, it says the excitation-contraction coupling. So written as is, it involves the excitation. It involves the neuron, I'll say the neuron, but of course it involves the neurotransmitter. So for this to occur, it must be a change in membrane potential. So that electrical current has been generated. Of course, we get that active potential here at the sarcolemma, and that active potential is then propagated along the sarcolemma, and intracellular calcium ion levels, they rise to trigger contraction. So the way this occurs was first by the active potential arriving at the axon terminal at the neuromuscular junction. Acetylcholine was released, and of course it became bound to its chemically gated sodium ion channels. So their ion permeability of the sarcolemma changed. Depolarization occurred, and that end plate potential, that depolarization event, caused the active potential. So as that active potential traveled across the sarcolemma and the T-tubules, because of that occurring, I repeat, the T-tubules is that active potential was allowed to get deep within the muscle fiber itself. So what happened there is that voltage-gated calcium channels then released calcium. So as long as that calcium that was released, meaning rise to levels that it has to be at, meaning put it this way, at low intracellular calcium levels, and we're here now in the muscle class, at low intracellular calcium levels, the muscle is relaxed, and it will remain relaxed. So tropomyosin will physically block the active sites on actin, those myosin binding sites. So as calcium levels rise, all because of the change in membrane potential, because the voltage-gated calcium channels opening, those ions will bind to the regulatory sites on troponin, the T and C, causing it to change its shape, then roll away tropomyosin into the groove of the actin helix. So the tropomyosin, which is called that blocker, is removed when sufficient calcium is present. So once those binding sites have been exposed on actin, the events of cross-bridge cycling occur in rapid succession. So those thin filaments slide as calcium signals, and adequate ATP is present. So with each cycle, the myosin head takes another step by attaching to another actin site further along the thin filament. So the way this happens, to make it, I guess I'll say, even clearer, is that calcium that has been released from that sarcoplasmic reticulum exposes the binding sites on the thin filament. And it's because there, calcium has become bound to troponin, and then tropomyosin is pulled aside. As it's pulled aside, of course, those binding sites on the thin filament are exposed. So it allows the contraction cycle to occur. So what happens next is the myosin heads will bind to actin, forming cross bridges, connecting the myosin to actin. Next up, ADP, adenosine diphosphate, and an inorganic phosphate. They are released from the myosin cross bridge, and they pull that filament, this is a power stroke, toward the M line. So next up, a new ATP will bind to the myosin to break that cross bridge, to break that connection to actin. So to reiterate this, ATP binds to actin for a twofold reason. A, for the detachment of the myosin head, and B, to return myosin to that cocked position. And it's all because of that hydrolysis of ATP, taking what was adenosine triphosphate and dephosphorylating it and taking it back to being adenosine diphosphate. So as I stated, ATP splits, which provides that power to cock the myosin heads and store the energy for that next power stroke. So this will continue, of course, until it ceases, and that's all based upon acetylcholine and, of course, nervous stimulation. Please be sure that you all are able to provide at least 2 contraction cycles. This is on your test. So to make sure I review just a bit. The binding of the acetylcholine to its acetylcholine receptors at the neuromuscular junction. It opens those chemically gated ion channels that allow sodium and potassium to pass. So more sodium diffuses out of that cell than potassium out, and that membrane potential inside the sarcolemma becomes less negative. It becomes depolarized, becoming more positive, I say. Depolarization and generation and propagation of that active potential ignites the active potential by spreading to adjacent membrane areas and opening voltage-gated sodium ion channels. So the sodium enters and it follows its electrochemical gradient, and once a certain membrane potential, the threshold is reached. And of course, we elicit that active potential. It's generated. It is initiated. The active potential propagates. It moves along the length of that sarcolemma in all directions from the neuromuscular junction. It's like ripples in a pond by throwing a rock. So as it propagates, the local wave of depolarization and the active potential, it spreads to adjacent areas on the sarcolemma, opening more voltage-gated sodium channels. So normally normally what is restricted called sodium, meaning it can enter, it now diffuses into the cell following its electrochemical gradient. So nextly, repolarization occurs, restoring the sarcolemma to its initial polarized state, and then repolarization wave is that consequence of the change in membrane permeability. Those sodium ion channels close and the voltage-gated potassium channels open, and since the potassium ion concentration is so much higher inside the cell than the extracellular fluid, potassium diffuses out rapidly from the muscle fiber, restoring the negatively charged conditions inside. What I've just given you, of course, was that excitation part and how this occurs and is predicated on the nerve impulse. If there are any questions about this class, please let me know. Otherwise, I shall continue on. So as I continue here, we then got into energy sources for contraction. Energy is created in a multiple of ways. Or at least I'll say in a multiplicity of ways. One way, of course, is by way of creatine phosphate. For short bursts of energy, creatine phosphate is used. So what happens is when sufficient ATP is present, the enzyme within the mitochondrion promotes the synthesis of creatine phosphate, which stores excess energy in that phosphate bond. So when it is needed, meaning ATP is needed, that adenosine diphosphate is phosphorylated, and of course we're left with just creatine because that phosphate is used. Another way in which it occurs is by respiration, meaning it synthesizes many ATP. And of course, it is stored in skeletal muscles, typically by way of glycogen. So we have another protein called myoglobin, which is synthesized in muscle cells. So this is why muscle cells have that reddish brown color. The skeletal muscle cells, I repeat. So myoglobin does bind to oxygen and has a greater attraction for oxygen than even hemoglobin. So myoglobin, it temporarily stores oxygen and increases the amount of available oxygen to our muscle cells. This supports aerobic respiration, you all, and the oxygen storage here in myoglobin is important because blood flow may decrease when the contraction of muscles compresses the blood vessels. With oxygen debt, which is next, this happens when those muscles, the skeletal muscles, are used more strenuously. So these systems, they may not be able to support enough oxygen to sustain those aerobic actions of respiration. So under these conditions, the muscles may be limited, I say limited only to the anaerobic reactions, to produce adenosine triphosphate. So here, this shift in metabolism, of course, is referred to the anaerobic threshold or the lactic acid threshold. So as I say it this way, it's that thing called glycolysis, meaning by way of the anaerobic reactions, that breaks down glucose, meaning the splitting of glucose, to yield those two pyruvic acids. So when this happens, meaning as oxygen supply is low, the pyruvic acid reacts to produce lactic acid, which enters the bloodstream and eventually reaches the liver. So I'll get now to oxygen debt, or that excess post-exercise oxygen consumption. So as I mentioned this, it must be repaid. So oxygen debt also reflects the oxygen required to restore blood and tissue oxygen levels to pre-exercise levels. So next up, I'll get to muscle fatigue. So this is that muscle exercise that is persistent, or at least for a prolonged period of time. It may lose its ability to contract, which is causing muscle fatigue. So also keep in mind that heat production occurs by way of our skeletal muscles. So the muscle tissue, it constitutes such a large proportion of total body mass. So that it is a major source of heat. I'll next get to the threshold stimulus. I say threshold stimulus because this is what must be reached to generate the active potential. So that is what results in that, of course, impulse that spreads throughout the muscle fiber and releasing enough calcium ions from that sarcoplasmic reticulum to activate cross-bridge cycling and, of course, contract the fiber. So that single active potential is what will release acetylcholine to bring the muscle fibers to threshold, generating the impulse. And it is at the approximate negative 55 millivolts. So now I'll get to the recording of a muscle twitch, as seen at figure 9.14. As you see the muscle twitch, pay attention to that latent period. It's during that latent period that when you have the events occurring that's nervous or that excitation portion. So it will always be a latent period by way of which you're having what has occurred here by way of that motor neuron before you get any contraction. So I'll put it this way. First will be the excitation portion, that latent period, and then that period of contraction, and ultimately that period of relaxation. So as you see this, I'll say that very same is the case. So muscle hypertrophy occurs because of the use of skeletal muscle. Or just put it this way, by exercising. Forceful exercise causes the muscle to get larger, and it's called, of course, the increase in size and strength. That is muscle hypertrophy. Nextly, I'll get to muscle atrophy, the decreasing in size and strength. So muscle atrophy begins, and as it does, it's because of, I guess I'll say, the lack of use. So since a person walks, moves their arms, and is just generally active, I would say those muscles remain to be in a form of muscle tone. However, if he or she were to become injured and cannot move anymore, what will then occur, of course, is that connective tissue would replace. Connective tissue would replace what was once muscle, and it's all due to muscle atrophy. Hey, buddy, how are you doing? Good to see you. Daddy's recording a video. I'm recording a video. Next up is smooth muscle. So with smooth muscle, it's wholly different from skeletal muscle. Skeletal muscle, as opposed to smooth muscle, is multinucleate. Smooth muscle has only one central nucleus. Only one central nucleus. So that one nucleus that's located in smooth muscle is one cube. Moreover, it's not striated. It has no myofibrils. So it looks smooth, and there are no transverse tubules. So smooth muscle contracts. As it does contract, it is slower to contract. So as it is slower to contract, I mean that it's contracting slower, and it is also slower to tire. So as smooth muscle contracts only by stimulation of a neuron or certain hormones, it is composed of those sheets of those spindle-shaped cells that are held in close contact by gap junctions. So when one cell of smooth muscle is stimulated, the impulse is conducted over its surface, and it may excite adjacent cells. So smooth muscle also in it that can stimulate other cells, it exhibits rhythmicity. As I state that smooth muscle exhibits rhythmicity, it's able to contract in a wave-like motion referred to as peristalsis, meaning that pattern of the spontaneous repeated contractions. So the peristaltic action I refer to here is what moves foodstuffs through the digestive tract, or even, of course, through what is called the ureters, by way of the kidney to the urinary bladder, meaning urine, you all. So I say again, smooth muscle exhibits conduction, the impulses being going from cell to cell, and rhythmicity with those rhythmic contractions. So from here, I'll move on to smooth muscle contraction. So with smooth muscle contraction, the mechanism it's similar. However, smooth muscle has no troponin, and in its place, smooth muscle has the protein called calmodulin. Yes, buddy?
That's my bike.
It is, but you're kind of small for that bike. Ride your other bike, please. So acetylcholine, the neurotransmitter that is in skeletal muscle, as well as norepinephrine, are what affect smooth muscle. The effect is going to be different, meaning each of those neurotransmitters, being acetylcholine and norepinephrine, they stimulate contraction in smooth muscle. However, it also inhibits contraction in other smooth muscle. So I'll say the effect of these neurotransmitters class depends on the receptor. So yes, I did say that each of those neurotransmitters can stimulate contraction in some smooth muscle and inhibit contraction in other smooth muscle. This goes all the way back to where we're getting to in subsequent chapters, being the autonomic nervous system. So I'll say it this way, that it's a number of hormones that affect smooth muscle by stimulating it or inhibiting it. Meaning some will stimulate contraction, some will inhibit contraction, or even alter the degree of which it will respond to a neurotransmitter. For example, during the later stages of childbirth, oxytocin, that hormone, is released, and it stimulates those smooth muscle wall contractions of the uterus. The stretch of smooth muscle can also contribute contractions. Particularly, I say this is with visceral smooth muscle and the wall of hollow organs such as our intestines and urinary bladder. So by way of our intestines and urinary bladder, meaning when partially digested food stretches the wall of the intestine, those contractions move the contents further along the intestines. So again, I'll reiterate that smooth muscle is slower to contract and relax than skeletal muscle. Yet, smooth muscle can forcefully contract longer with the same amount of ATP. So unlike unlike skeletal muscle, smooth muscle cells can change length without changing tautness. So this ability is what enables smooth muscle in the stomach and the intestinal walls to stretch as these organs filled, yet maintain constant pressure inside those organs. Next up is cardiac muscle. So I'll let alone cardiac muscle by and large because, of course, it's dealt with in anatomy 2. And this is Human Anatomy and Physiology 1. What I will say is cardiac muscle is found in the heart, only in the heart. It is composed of striated cells that are joined end to end, and they form that interconnected branching network. And there they are intercalated discs at gap junctions, and it has one nucleus per cell. It has a well-developed sarcoplasmic reticulum because calcium is majorly important. It has an amazing system of T-tubules, transverse tubules, and many mitochondria. So next of which I'll say opposing ends of cardiac muscle are connected again by bands called intercalated discs. So with these junctions, they have desmosomes here as well as gap junctions. So those intercalated discs help those cells transfer the force of contraction from cell to cell. As cardiac muscle contracts, it contracts wholly. It's a functional synchrony. And I say that because the network responds in this all or none contraction manner. So cardiac muscle is self-excitable and rhythmic. It has a pattern of contraction and relaxation, and it repeats. So it generates the rhythmic contraction of the heart. Cardiac muscle also becomes non-responsive after stimulation until the contraction ends. So this gets back to the refractory period I'll get to, of course, with the nervous system. So therefore, it doesn't have tetanic contractions or sustained contractions, not in the heart at all. What I'll say now, class, is please make sure you all study very well for the exam. If you have any questions about this chapter being chapter 9, please let me know. In the meantime, please take the time to have taken notes and review your notes and ask me any questions that you all may have. In the meantime, this has been your instructor, Scholar Huff. Take care of yourselves, class. Study very well. And if I can help you with anything, again, like I say, please do not hesitate to let me know.